Hello, everyone. So we will be starting with chapter 9 of Illustrated Textbook of Pediatrics. That is genetics. Features of the genetic basis of diseases are the Human Genome Project resulted in the first publication of the human genome sequence in 2001. It is now estimated the human genome contains 20,000, 25,000 genes, although the function of many of them remains unknown. Greater diversity and complexity at the protein level is achieved by alternative messenger RNA splicing and post-translation modification of gene products, microarray techniques, and high throughput sequencing are increasing the volume and speed of genetic investigations and reducing their costs, leading to a greater understanding of the impact of genetics on health and disease. Access to genome browser database containing DNA sequence and protein structure has greatly enhanced progress in scientific research and the interpretation of clinical test results. Genetic databases are available on thousands of multiple congenital anomaly syndromes, on chromosomal variations and disease phenotypes, and on all Mendelian disorders. Clinical applications of these advances is available to families through specialist genetic centers that offer investigation, diagnosis, counseling, and antenatal diagnosis for an ever-widening range of disorders. Gene-based knowledge is entering mainstream medical and pediatric practice, especially in diagnosis and therapeutic guidance, such as for the treatment of malignancies. Genetic disorders are common, with 2% of live-born babies having a significant congenital malformation and about 5% a genetic disorder. Burdensome to the affected individual's family and society, as many are associated with severe and permanent disabilities. Genetically determined diseases include those resulting from chromosomal abnormalities, the action of a single gene, that is Mendelian disorders, unusual genetic mechanisms, interaction of genetic and environmental factors, which are polygenic, multifactorial, or complex disorders, which include epigenetic influences on gene expression from early in life. Now it's chromosomal abnormalities. Genes are composed of DNA that is wound around a core of histone proteins and packaged into a succession of supercoils to form the chromosome. Human chromosome complement was confirmed as 46 in 1956. The chromosomal abnormalities in Down, Klinefelter, and Turner syndromes were recognized in 1959, and thousands of chromosome defects have now been documented. Chromosomal abnormalities are either numerical or structural. They occur in approximately 0% of spermatozoa and 35% of mature oocytes and are a common cause of early spontaneous miscarriage. The estimated incidence of chromosomal abnormality in live-born infants is about 1 in 150. They often cause multiple congenital anomalies and cognitive difficulties. Acquired chromosomal changes play a significant role in carcinogenesis and tumor progression. This figure 9.1 shows ensemble genome browser. The images show part of chromosome region 22q1 involved in 22q1 deletion syndrome, that is de Jour syndrome. Although only part of the commonly deleted region is shown, the image shows several genes that are deleted in 22q11 deletion syndrome. The online ensemble browser can be used to zoom in on specific areas showing the genes present in different chromosome regions and can also be used to show the gene sequence itself. Now, disorders of chromosomal number. First one, is Down syndrome, which is trisomy 21. This is the most common autosomal trisomy and most common genetic cause of severe learning difficulties. The incidence without antenatal screening in live-born infants is about 1 in 650 and increases with maternal age. Clinical features if not diagnosed antenatally, Down syndrome is usually suspected at birth because of the baby's facial appearance. Most affected infants are hypertonic and other useful clinical signs include a flat occiput, single palmar crease, incurved fifth finger, and wide sandal gap between the big and second toes. The diagnosis can be difficult to make when relying on the clinical signs alone, and a suspected diagnosis should be confirmed by a senior pediatrician before blood is sent for analysis. Parents should be informed that a test for Down syndrome is being performed. The result may take 1 to 2 days using real-time PCR or rapid fluorescence in situ. Hybridization is also found fish. Techniques. Parents need information about the short-term and long-term implications of the diagnosis. They are also likely at some stage in the future to appreciate the opportunity to discuss how and why the condition has arisen, the risk of recurrence, and the possibility of antenatal diagnosis in future pregnancies. It is difficult to give a precise long-term prognosis in the neonatal period as there is great individual variation in the degree of learning difficulty and the development of complications. Over 85% of infants with trisomy 21 survive to 1 year of age. Congenital heart disease is present in about 40% and is a major cause of early mortality, particularly atrioventricular canal defects. Duodenal atresia is another problem in the newborn period. However, in the UK, at least 50% of affected individuals live longer than 50 years. Parents also need to know what assistance is available from both professionals and family support groups. Counseling may be helpful to assist the family to deal with feeling of grief, anger, or guilt. Child development services will provide or coordinate care for the parents. This will include regular review of the child's development and health. Child with Down syndrome should be screened periodically for impairment of vision and hearing, hypothyroidism, celiac disease, and atlantoaxial instability. Cytogenetics, extra chromosome 21 may result from meiotic non-dysfunction, translocation, or mosaicism. Meiotic non-dysfunction in 94% of cases, in non-dysfunction trisomy 21, most cases result from an error at meiosis. The chromosome 21 pair fails to separate so that one gamete has 2 chromosome 21s and one has none. That will be shown in the figure later. Fertilization of the gamete with 2 chromosome 21s give rise to a zygote with trisomy 21. Parental chromosomes do not need to be examined. Incidence of trisomy 21 due to non-dysfunction related to maternal age. However, as the proportion of pregnancies, the older mother is small, most affected babies are born to younger mothers. Furthermore, meiotic non-dysfunction can occur in spermatogenesis so that the extra copy of chromosome 21 can be of maternal age. All pregnant women are now offered screening tests, measuring biochemical markers in blood samples and knuckle thickening on ultrasound, thickening of the soft tissue at the back of the neck to identify an increased risk of Down syndrome in the fetus. When an increased risk is identified, amniocentesis is offered to check the fetal karyotype. Non-invasive prenatal testing, NIPT, is now possible, in which cell-free fetal DNA is analyzed from maternal blood and is becoming part of routine screening in the UK. After having one child with trisomy 21, due to non-dysfunction, the risk of recurrence of Down syndrome is given as 1 in 200 for mothers under the age of 35 years, but remains similar to their age-related population for those over the age of 35 years. Now, this is the block of Down syndrome, figure 1. Figure 9.2a, characteristic faces seen in Down syndrome are posture due to hypertonia. Figure number 9.2b, single palmar freeze. 9.2c, pronounced sandal gap with wide space and often a deep fissure between the big toe and second toe. Characteristic clinical features of Down syndrome. Typical craniofacial appearances, round face and flat nasal bridge, upslanted palpebral fissures, epicanthal folds, a fold of skin running across the inner edge of the palpebral fissure, brush-field spots in iris, pigmented spots, small mouth and protruding tongue, small ears, flat occiput and third fontanelle, other anomalies, short neck, single palmar creases, incurved and short fifth finger and wide sandal gap between first and second toes, hypertonia, congenital heart defects in 40%, urinary atresia, Hirschsprung disease in less than 1%. Later medical problems, delayed motor milestones, learning difficulties, severity is variable, usually mild to moderate, but maybe severe. Short stature, increased susceptibility to infections, hearing impairment from sick uveitis media, 75% vision impairment from cataracts, 15% squint myopia is 50%. Increased risk of leukemia and solid tumors, less than 1%. Acquired hip discoloration, dislocation, and atlantoaxial instability. Obstructive sleep and apnea, 50 to 95, 50% to 75%. Increased risk of hypothyroidism, 15%, and celiac disease. Epilepsy, early onset, Alzheimer disease. Now the inheritance of non-dysfunction, it's showing inheritance of Down syndrome, it's showing non-dysfunction and Robertsonian translocation. Non-dysfunction shows parent and one of the parent has non-destruction at meiosis, one of the chromosomes and non-destruction meiosis, gametes with the 21 DNA, fertilization, and then offspring has trisomy 21. This one is Robertsian translocation carrier. One of the parents is translocation carrier, the other one is normal. Gametes form are translocation involving chromosome 21 and 14, which has been inherited from one parent. Offspring include normal translocation carrier, monosomy 14 not viable, disomy 14 not viable, monosomy 21 not viable, translocation Down syndrome. So it is more explained in detail. Translocation 5% when the extra chromosome 21 is joined onto another chromosome, usually chromosome 14, but occasionally chromosome 15, 22, and 21. This is known as Robertsonian translocation. This may be present in a phenotypically normal carrier, 45 chromosome, 2 being joined together, or in someone with Down syndrome and a set of 46 chromosomes, but with 3 copies of chromosome 21 material. In this situation, parental chromosome analysis is recommended because one of the parent may well carry the translocation in balanced form in 25% of cases. Now, the table shows risk of Down syndrome, live births, maternal age at delivery prior to screening, maternal age in years, and then risk of Down syndrome. In all ages, the risk is 1 in 650. At 20 years of age, it is 1 in 1530. On 30 years of age, is 1 is 900. 35 is 1 in 385. And 37, it's 1 in 240. And at 40, it's 1 in 110. And at 44, it is 1 in 37 live births. In translocation Down syndrome, the risk of recurrence is 10 to 15% if the mother is a translocation carrier and about 2.5% if the father is the carrier. If the parent carries the rare 21:21 translocation, all the offspring will have Down syndrome. If neither parent carries a translocation, 75% cases, the risk of recurrence is less than 1%. Mosaicism, some of the cells are normal and some have trisomy 21. This usually arises after the formation of the chromosomally normal zygote by non-dysfunction at mitosis, but can arise by later mitotic non-dysfunction in a trisomy 21 conception. The phenotype is sometimes milder in Down syndrome. Now, summary. Down syndrome, trisomy 21, national incidence about 1.5 per 1,000 infants, cytogenetics, Non-dissection, most common, related to maternal age translocation. One parent may carry a balanced translocation, or mosaicism is very rare, about 1%. Presentation, antenatal screening, prenatal diagnosis, or clinical presentation confirmed a chromosomal analysis. Immediate medical complications, increased risk of duodenal atresia, congenital heart disease. Clinical manifestations, as shown in the box. Now, this is Edwards syndrome and Patau syndrome. Edwards syndrome is trisomy 18, and Patau syndrome is trisomy 13. Edwards syndrome and Patau syndrome, clinical features of Edwards syndrome trisomy 18. It is low birth weight, prominent occiput, small mouth and chin, short sternum, flexed overlapping fingers as shown in the figure, rocker bottom feet, cardiac and renal malformations. As shown in the figure, overlapping figures in Edwards syndrome. Now the clinical features of Patau syndrome, that is trisomy 13, structural defect of brain, scalp defects, small eyes, microphthalmia, and other eye defects, cleft lip and palate, polydactyly, and cardiac and renal malformations. Now, Edward syndrome, trisomy 18, and Patau syndrome, trisomy 13, although rarer than Down syndrome, 1 in 8,000, 1 in 48,000 live births, respectively. Particular constellations of severe multiple abnormalities suggest these diagnoses are birth. Most affected babies die in infancy, as shown in the boxes, but extended survival is possible. Diagnosis confirmed by chromosomal analysis. Many affected fetuses are detected by ultrasound scan during the second trimester of pregnancy, and diagnosis can be confirmed antenatally by amniocentesis and chromosome analysis. Can also be diagnosed on non-invasive prenatal testing. Recurrence risk is low, except when the trisomy is due to a balanced chromosome rearrangement in one of the parents. The next one is Turner syndrome, which is 45X. Turner syndrome usually results in early miscarriage, more than 95%, and it's increasingly detected by ultrasound antenatally when fetal edema of the neck, hands, or feet, or cystohygroma may be identified. In live-born females, the incident is about 1 in 2,500, as shown in the figure 9.6, and box shows the clinical features of Turner syndrome. Clinical features of Turner syndrome includes lymphedema of hands and feet in neonate, which may persist, spoon-shaped nails, short stature, a cardinal sign, neck webbing, a thick neck, wide carrying angle, cubitus valgus, widely spaced nipples, congenital heart defects, particularly coarctation of the aorta, delayed puberty, ovarian dysgenesis, resulting in infertility. Although pregnancy may be possible with in vitro fertilization using donated ova, hypothyroidism, renal anomalies, pigmented males, recurrent itis media, and a normal intellectual function in most cases. This figure shows the woman on the left has marked short stature, but no other clinical features. Whereas the female on the right has neck webbing, has received growth hormone, and is 150 centimeter in height. Although short stature may be the only clinical abnormality in children, treatment is with growth hormone therapy, estrogen replacement for development of secondary sexual characteristics at the time of puberty, but infertility persists. In about 50% of girls with Turner syndrome, there are 45 chromosomes with only one X chromosome. The other cases have a deletion of the short arm of one X chromosome and isochrome that has two long arms but no short arm, or a variety of other structural defects of one of the X chromosome. The presence of a Y chromosome sequence may increase the risk of gonadoblastoma. The incidence does not increase with maternal age, and risk of recurrence is very low. Klinefelter syndrome, 47XXY. The disorder occurs in about 1 to 2 per 1,000 live-born males. For clinical features, the box will be shown. Recurrence risk is very low. Structural chromosomal anomalies, reciprocal translocation, and exchange of material between two different chromosomes is called reciprocal translocation. When this exchange involves no loss or gain of chromosome material, the translocation is balanced and usually has no phenotypic effect. Balanced reciprocal translocation are relatively common, occurring in 1 in 500 of the general population. A transformation that appears balanced or conventional chromosome analysis may still involve the loss of few genes or the disruption of a single gene at one of the chromosomal breakpoints and result in an abnormal phenotype, often including cognitive difficulty. Studying the breakpoints in such individual has been one way of identifying the local location of specific genes. Unbalanced reciprocal translocation contains an incorrect amount of chromosome material and often impair both physical and cognitive development, leading to dysmorphic features, congenital malformations, developmental delay, and learning difficulties. When recognized in a newborn baby, the prognosis is difficult to predict, but the effect is usually severe. The parent's chromosome should be checked to determine whether the abnormality has arisen de novo or as a consequence of parental rearrangement. Finding a balanced translocation in one parent indicates a recurrence risk for future pregnancies, so that antenatal diagnosis by chorionic villus sampling or amniocentesis should be offered, as well as testing relatives who might be carriers. Deletions are another type of structural abnormalities. Loss of part of a chromosome usually results in physical abnormalities and cognitive impairment. Deletion may involve loss of terminal or an interstitial part of the chromosome arm. An example of a deletion syndrome involves loss of a tip of a short arm of chromosome 5, hence the name 5p or monosomy 5p, because affected babies have a high-pitched mewing cry in early infancy. It is also known as try-do chat. Parental chromosome should be checked to see if one parent carries a balanced chromosome rearrangement. The clinical severity varies greatly depending on the extent of the deletion. It is now possible to specify the genes involved in chromosomal deletion, as molecular methods are replacing standard cytogenetic investigation. Deletion of band Q11 or chromosome 22, i.e. 22q11, can be associated with the range of phenotypes and the velocardiofacial syndromes. Williams syndrome is another example of a microdeletion syndrome due to loss of chromosome material at band Q11 on the long arm of chromosome 7. We will show the figure. Now, the duplication gain of structural material can also lead to congenital malformation and intellectual impairment, although duplications are often better tolerated than deletions. An example of duplication syndrome in partial trisomy of 17p, duplication can range from being submicroscopic to being large enough to be visible on a karyotype. If it involves duplication of the PMP22 gene at 17p12, then the patient will have a form of Charcot-Marie-Tooth disease, peripheral neuropathy, in addition to other features resulting from the abnormality. Now we should read the box, that is 9.5, clinical features of Klinefelter syndrome. Number 1 is infertility, most common presentation, hypogonadism with small testes, pubertal developmental may appear normal. Some males benefit from testosterone therapy. Gynecomastia and adolescence, tall stature, intelligence usually in the normal range, but some have educational psychological problems. Testing for microscopic copy number variants. An increasing number of syndromes are now known to be due to chromosomal due to chromosome deletions or duplications too small to be seen by conventional cytogenetic analysis. Submicroscopic deletions can be detected by FISH studies using DNA probes specific to particular chromosome regions. FISH studies are useful when a specific chromosome deletion is suspected. Newer techniques are beginning to supersede the karyotype in FISH. Array comparative genomic hybridization microarray can be used to check the chromosome for structural rearrangements larger than 150 kb in size, as shown in the figure. We will discuss the figure later. The advantage of microarrays is that no specific target is required for the test to be effective. Many of the newly emerging submicroscopic copy number variants have overlapping clinical features, and non-targeted texting is the most effective way to identify these. One disadvantage of array comparative genomic hybridization is that information provided can be unhelpful if the test reveals a copy number variant of uncertain significance. In addition, microarrays only provide quantitative data and cannot be used to check for structural rearrangements. For example, Robertsonian translocation. Information provided by microarray often requires discussion with the clinical geneticist. Mendelian inheritance. First, we will discuss these cytogenetics, analysis techniques, karyotype resolution, and microscopic, submicroscopic rearrangements. Fluorescence in situ, which is FISH, specific to target, microscopic and submicroscopic rearrangements are present. Comparative genomic hybridization array, which is microarray, that can check the chromosome for structural rearrangement, which is larger than the resolution is 150 kb. So microscopic, it's seen, and in karyotype, it's seen in microscopic and rearrangements. So the next one is the picture showing fluorescence in situ hybridization, demonstrating a microdeletion on chromosome 22 associated with DiGeorge syndrome. Hybridization signals are seen on one chromosome 22, but not on the other because of the presence of a deletion. And this also shows array comparative genomic hybridization microarray result from patient with 22q11 deletion. There is a reduction in the ratio of patient control sequences from within band 22q11 on the long arm of chromosome 22. Now, the Mendelian inheritance. Mendelian inheritance described by Mendel in 1866 from work on garden peas is the transmission of inherited traits or diseases caused by variation in a single gene in a characteristic pattern. These Mendelian traits or disorders are individually rare, but collectively numerous and important. Over 6,000 have been described so far. For many disorders, the Mendelian pattern of inheritance is known. If the diagnosis of a condition is uncertain, its pattern of inheritance may be evident on drawing a family tree pedigree, which is an essential part of genetic evaluation. Now, as shown in the figure is an example of pedigree symbols. It shows a circle, empty circle, empty square, and empty diamond, which is unaffected, female, male, and sex unknown. And then now the affected are shaded. They're affected female, male, and sex unknown. There are heterozygotes, carriers autosomal recessive inheritance, and female and male are half shaded. Female heterozygote carrier in X-linked inheritance. Okay. And when male and female are joined with one line, it is mating. And when they're joined by two lines, it is consanguineous mating. And the examples are shown mating with mating with two offsprings, and then another pedigree showing an abortion, and then dizygous twins are shown like this. Two arrows and monozygotic twins are shown by a triangle between them, and deceased are shown with a line crossing a square or a for a male and a sorry, a circle for a female, and index case is shaded and with an arrow, and for unaffected female is with a circle and which is not shaded as shown, and it is number 4 for the number. Autosomal dominant inheritance, this is the most common mode of Mendelian inheritance that is shown in box 9.6. Autosomal dominant conditions are caused by alterations in only one copy of a gene pair, i.e., the condition occurs in the heterozygous state despite the presence of an intact copy of the relevant gene. Autosomal dominant gene are located on the autosomes chromosome 1 to 22, and so males and females are equally affected. Each child from an affected parent has a 1 in 250% chance of inheriting the abnormal gene that we show in the figure. This appears to be straightforward, but complicating factors include the following factors: variation in expression. Within a family, some affected individuals may manifest disorder mildly and others more severely. This may be a result of variation at other genes, environmental effects, or sheer chance. Non-penetrance refer to a lack of clinical signs and symptoms in an individual who has inherited then the abnormal gene. An example of this is autosclerosis, in which only about 40% gene carriers develop deafness. That we shown in the figure ahead. De novo mutation. Mutation is classed as de novo if it does not affect either parents. It may be due to a new mutation in one of the gametes, leading to the conception of the affected person. This is the most common reason for absence of family history in dominant disorders. For example, about 80% of individuals with achondroplasia have unaffected parents. The risk of new single point mutation increases with paternal age. Paternal mosaicism, very occasionally a healthy parent harbors the mutation only in some of their cells. For example, in their gonads, this can account for recurrences of autosomal dominant disorder in siblings born to apparently unaffected parents. It has been described in congenital lethal osteogenesis imperfecta. Non-paternity if the apparent father is not the biological father. Homozygosity, in the rare situation where both parents are affected by the same autosomal dominant disorder, there is a 1 in 4 risk that a child will be homozygous for the altered gene. This usually causes a more severe phenotype, which may be lethal, as with achondroplasia. Knudsen two-hit hypothesis is some autosomal dominant conditions related to cancer susceptibility follow Knudsen two-hit hypothesis. Both copies of the gene need to be mutated for a malignancy to occur. If a person is born with only one working copy of the gene in every cell in his or her body, then only one further mutation event needs to occur for both copies of the gene to be inactivated. The chance of this happening is much greater than the chance of 2 successive mutations occurring in someone who starts life with 2 functional copies of the gene. The susceptibility to cancer is therefore inherited in a dominant fashion, but the development of cancer within a cell can be thought of as a local event within the individual, so that not every person who inherits susceptibility will necessarily develop a malignancy. An example in pediatrics is mutation in the retinoblastoma RB gene. If a child inherits the susceptibility, that is a mutation in one copy of RB gene, then a tumor will occur if the second hit occurs on the working copy in a cell of the relevant type, so that the child inheriting a mutation will often have a tumor in both eyes, but approximately 10% will escape with neither eye affected. These are the autosomal dominant Inheritance examples. Examples of autosomal dominant disorder: achondroplasia, Ehlers-Danlos syndrome. This is a family of disorders rather than a single condition. Familial hypercholesterolemia, Huntington disease, Marfan syndrome, myotonic dystrophy, neurofibromatosis, Noonan syndrome, osteogenesis imperfecta, or autosclerosis, polyposis coli, and tuberous sclerosis. Now, the autosomal dominant inheritance. Parents' one gene is affected and offsprings are 50% affected and 50% are normal. Typical pedigree of a autosomal dominant disorder: one parent is affected, and these are the 50% chances of getting affected and 50% chances of normal individuals as shown in the pedigree. Summary: autosomal dominant inheritance. Most common mode of Mendelian inheritance. Affected individual carries the abnormal gene on one of the pair of autosomes. There is a 1 in 2 chance of inheriting the abnormal gene from affected parent, but there may be variation in expression, non-penetrance, no family history, new mutation, parental mosaicism, nor paternity, or homozygosity, which is rare. Now, in figure 9.10, example of non-penetrance 1 and 3 have autosclerosis, and in number 1, the female has autosclerosis, and 2 have autosclerosis. Okay. Number 3 and 2 have autosclerosis. Number 2 have normal hearing but must have the gene. A new mutation event is most unlikely to arise independently for a second time in the family. The gene is non-penetrant into number 2. Autosomal recessive gene. An infected individual is homozygous for the mutant allele in the gene. Sometimes there is a different mutation on each copy of the gene. For example, cystic fibrosis, and the affected individual is a compound heterozygote. In either situation, they will have inherited an abnormal allele from each parent, both of whom will usually be unaffected heterozygous carriers. For a couple with both parents being carriers, the risk of any child being affected, male or female, is 1 in 4, 25%. As shown in the figure, all offspring of an affected individual will carry the condition. If an affected individual has children with an unaffected carrier, then each has 50% chance of being affected.